All right, hello everyone. I hope you enjoy your lunch. I think we need to start so we can finish on time. It's Friday afternoon. Some of you may need to go home or far away. I'm Pavlos Protopappas. I am the Scientific Program Director of the Institute, and I will be introducing the speakers for the afternoon. We have 3 speakers in the afternoon, followed by a panel discussion. The afternoon focus will be now on actually how the brain works, how it's wired. As I said, we have 3 speakers, Jeff, followed by Nancy, and then Andreas. Let me first introduce Jeff. Jeff is a professor of molecular and cellular biology here at Harvard, and he has done great contributions how the brain is connected with the Connectomics project, and I would not take more of his time. Jeff, it's your turn. Thanks.
Because this is about brains and computers, I think it's important to state at the outset that it's true that both computers and brains take information from the world and use this information to solve problems. But at least from my perspective, at the moment there are really vast differences between the way computers and brains work. After all, this is a cross-section of a human brain. This is a kind of nanotechnology that far exceeds anything humans have ever built. Low energy, you heard this from Dave Cox this morning, it's 3 pounds, supercomputer with 20 watts of power. That's magic relative to what we do with our megawatt computer arrays. It's hyperparallel. We don't do anything like this in computers. We can't wire computers to be as parallel as these are. And it's got a very slow clock speed compared to computers. It's kilohertz, not gigahertz. And it has analog and digital processing that's interleaved. And there are trillions of functionally important elements, such as synapses, and they come in many kinds. And the scale at which it is designed is at the nanometer scale. And we have little or to no idea how it actually works. So we're not in a really great position to be reverse engineering computers because we have no idea how this works. And I would say, should we wish to have computers that emulate brains, it might be a good idea then to understand first at a structural sense what a brain is actually made of, to know the features of brains that allow these amazing properties to exist. And today, what I'd like to do is just briefly emphasize some of them that are not part of modern day computers, but are part of all brains, especially human brains. And the first is that brains have a great structural and functional diversity that far exceeds anything in computers. This is, again, a cross-section of a brain now in cartoon, but even at the centimeter scale, this is the front, by the way, that's the back. It's the top of your head. You remove the frontal part of the brain, you'll have trouble planning. Part of the left temporal lobe and parietal lobe, you'll have trouble with speech. The occipital lobe, if that's damaged, you'll have trouble seeing. Balance is in the cerebellum, breathing is in the brainstem, walking is in the spinal cord. There's this division of labor, and not only do each of these parts have different functions, but they're different from each other. They have fundamentally different kinds of things going on in them as far as we know. That's at the macro level, but at the micro level, at the level of a microscope, a light microscope, there is another kind of tremendous diversity. And you see this if you go to a part of the brain where it's easy to see lots of cells. And this menagerie of shapes here are all the cell types in the retina in 2001. This shows cells that are interneurons and cells that have various other functions, bipolar cells, amacrine cells, and ganglion cells. These are the cells that leave the retina and send the information into the visual thalamus. And from there, the information goes to the cortex. And these different shapes are not just arbitrary. These shapes connote cells that have different functions. Some are very important for motion sensing, others are very important for fine vision, others for color. And this menagerie has gotten much larger, and there is a great effort right now to try to get the complete set of cells. And so someday we will have the complete set of cells of different types of cells. Each of these cells is iterated over and over again in the retina, and that will be a great triumph. That number right here, there's about 39. I think that number will end up being well over 100. That will be wonderful if you're interested in the retina, but all you have to do is go to the thalamus and you find a whole different menagerie of cells, or go to the cerebral cortex, and again, an extraordinary range of cells, the cerebellum, the amygdala, the brainstem, spinal cord. Every part of the brain has this extraordinary explosion of different types of cells. This kind of diversity just does not exist in man-made machines. And then finally, there is the nanoscale. Brains have important structural diversity in the macro, micro. I've already told you about nano. And I'm just going to give you a very quick primer in how what we teach neurobiology students about brains. You have the brain has a rind, the cortex has a rind called gray matter, where the nerve cells are, and then nerve cells connect to cells in other parts of the gray matter by sending myelinated axons from one region to another, and that's why the white matter, which is painted pink here for confusion, or orange, is white, or whitish in color. So there's this division of labor between gray matter and white matter, and then this is about 1 centimeter. If we just zoom up on one little piece of cortex, it's filled with these crazy-looking cells. Neurons don't look like any other cells in the body. They have a cell shape that's about the size of a typical cell anywhere else you might find, but coming out of those cells is this extraordinary explosion of branches. All of these branches are called dendrites. That's the receptive side of the cell. It's the antennae that this cell uses to receive information from other cells. This cell is receiving information from thousands or more friends, if it was Facebook, I guess, that are talking to it. And then if it hears enough synchronously interesting information, then that cell sends that message out its one axon, and that axon goes far-flung, far away, branches to thousands of other friends. And this is one cell of 100 billion or so like this. This is called a pyramidal neuron with a big apical dendrite. This is not just a random wiring of this dendritic tree. This is the way this particular cell type looks. If we zoom up, this is a millimeter. And this cell's axon can go 1 centimeter or 10 centimeters even. Really quite remarkable how long these cells are. If we zoom up and just look at the dendrites of this cell, we find another division of labor. The cell has these little spines, and the spines are where the cells receive information from other axons of other cells. These are the receptive places for excitation of these cells, and most of the inhibition, synapses on these cells that try to shut this cell up, are made on the shafts of the dendrites. So there's another division of labor there. And if we want to zoom up and look at a single spine, then we really go beyond the realm of light microscopy and have to start looking in the electron microscope. And here is a picture. I'll color this in for you. This is a picture from a serial electron microscopy data set of the brain. And this piece is a dendrite with one of these necks and a spine. And this little stuff in here is called a spine apparatus. And on the other side, just facing it, is this cross-section of something with a bunch of little vesicles in it. Those are synaptic vesicles. They have neurotransmitter. And this is the axon that's talking to this spine. And this is one of thousands of spines that this neuron, and that's just one dendrite of that neuron, has. So this is the synaptic connection, and the flow of information is from the axon to the dendrite. And so there's a big data problem here. There are lots of tools that begin to give us a sense of the activity of the brain and the organization of the brain at this relatively macroscopic level. One technique is functional magnetic resonance imaging, where the very best resolution of that technique is about a cubic millimeter per voxel. It measures blood flow locally in the brain, and it is remarkably good at telling us parts of the brain that are active at particular times during the time a human being is doing a task. At this resolution, if you want to see the brain so that you can see every synapse and perhaps every synaptic vesicle, then you're using voxels, three-dimensional pixels, that have a resolution of about 480 cubic nanometers. You all appreciate, I'm sure, that 480 cubic nanometers is a smaller voxel than 1 cubic millimeter, but unless you take out your calculator, you will probably not be aware that it's 2,000 trillion times smaller. That is, in one of these voxels, there are 2,000 trillion of these voxels, 2 petabytes, 2,000 terabytes of data, just to fill up 1 cubic millimeter of brain. And the IARPA project that we're on is trying to do the lofty goal of getting all the wiring in 1 cubic millimeter, which you might say $100 million for things like that just seems crazy, but that's a huge amount of data. That's 1 billion synapses. The next thing I want to point out is that a difference between the brain and most of the computers we make is that the structure of each brain derives from a combination of hereditary and experiential influences. And it's not just the function, but the structure, the physical structure of the brain is impacted by both what comes in through heredity, through the genes, and what is tuned based on the world the brain finds itself in. So biologists are very happy about the fact that genetic information, the genome, generates instructions to build the structures of the body, including the brain, and those structures generate the function of parts of the body, including the brain. But with the brain of animals like us, other mammals, other terrestrial vertebrates certainly, a certain amount of the structure could not have come from the genes. It had to have come from something related to experiencing the world. For example, most of you now can communicate with your thumbs on these little devices. Those devices were invented after many of us were born. So there's no way we have a gene for that. We must have some other way of generating, and once we've learned that, it's kind of stuck, just like riding a bicycle is stuck. So there is this kind of experience and use of the brain somehow gives rise to long-term stable changes that once they're in there, generates changes in function. And this is a loop, and loops are horrible. Biologists hate loops because unlike, you knock out a gene, you change the structure, you change the function, here it's a much messier thing. So we like to ignore it, but the brain, molecular networks, and certainly this is one of those classic loops. And this is the loop of memory, where information comes in and it turns into some physical state that lasts for very long times, an engram. So you could call this engrammatization. That's just a made-up word, but that would be what this is about. And my own personal interest is in this mechanism because I think this is where all cultural evolution comes from. Brains that evolve now, not simply based on changes in genes, but based on experience. So how do we see the nervous system at the level where we could see these kinds of structures? We have to see each and every nerve cell. These are not changes. Engrammatization doesn't mean that you're going to connect your arm to your brain in a different way. It's just the fine structure of those wires that are going to change. And in order to see that, I guess our view has been that maybe we would like to see every single nerve cell at the same time. And we built transgenic mice, which we call rainbow mice, where every single nerve cell is a different color. And this is just a stack of images through brainstem. And each of these little things is a wire. These are axons. And you can follow them easily back and forth, especially the ones going straight into the plane as we focus up and down. And these are cell bodies where they are encrusted with synapses, in this case all from one axon, and this is a cell where half of the synapses come from one axon and half from another. These are calyx inputs in the auditory brainstem. And these things running very fast sideways are axons that are running diagonally in data sets. This gives us some idea of the structure of the brain, and I just tell you a brief story about this. In the peripheral nervous system, you can see these wires very far that are labeled with rainbow. And when we first saw this, we were very proud of ourselves because we had done something quite remarkable until someone pointed out to me that this had already been done by computer scientists who realized that if you want to follow a wire over a long distance, color is a very good idea. So this is a kind of convergent evolution where our mice are looking more like computers slightly. If we zoom up on a little area like this, you can see that there really is a lot of color richness here. You can easily follow these axons long periods. And when these axons end up in muscle, these are axons that cause muscles to twitch. You can see the synapses these axons make in muscle. So here's a rotating little piece of a muscle, and these pretzel-shaped objects are the neuromuscular junctions, the place where axons touch muscle fibers. And even without tracing them all the way back to the spinal cord, I'm certain that this neuron and this neuron are different because they're different colors. And I'm also certain that these two that are purple are the same. And you can see that lots of different axons from different neurons are converging on this one little part of the muscle. And just one other thing I just want to emphasize for a second is that there's one axon coming to each muscle fiber. The muscle fibers are running vertically here, and they're cylindrical in shape. And you can see these things look like they're clasping cylinders. They're not labeled, the muscle fibers, but there's one of these on each muscle fiber. You trace a muscle fiber, only one axon talks to each adult muscle fiber. So we can now not just do a small region, but we can do the whole muscle. We can map out the entire wiring diagram of a muscle. And here's one of the axons going into a very small muscle that wiggles the ear of a mouse. And here's another axon in the same muscle, and another axon in the very same muscle. And you see these axons are kind of intermixed. These little things are those pretzel-shaped objects. And here's another one. Don't worry, there's only 15. This isn't going to go on too long. Zhu Lu, was the first author, insists that I show it this way because this was so much work to do that he said if I just showed it all at once, people wouldn't be impressed with how much work he had to do. So I have to show it this way by contract with him. But it does give you a sense of how some of them have lots of branches, others have fewer. And they are arbitrary. They seem very, very arbitrary where they go. But every muscle fiber only gets one branch at the end, and we have only one more to go here. This is one mouse, the left ear of one mouse. Great. So that's exactly what we did. We did the same thing on the other ear, and then we did 6 animals. Because the ears are mirror symmetrical. The body is mirror symmetrical. So is this mirror symmetrical? Every instantiation was completely unique. So that's already a warning, a ding, 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 about what to make of a wiring diagram. The other thing that's even more shocking is this was filled with suboptimality. Lots of things that didn't make any sense at all, and that I think were probably just not optimal. And let me give you an example here. This is one of Zhu Lu's reconstructions, and only one of the axons is labeled a different color here so you can see it clearly, and this region is just zoomed up here. All the axons send a branch up this black arrow direction except 2. This blue axon, which is an axon that bifurcates here, and this red axon in this particular muscle decides to go this way and not go that way. And who am I to judge? I mean, maybe there's a good reason to do that, but if you follow this axon, it makes a hairpin turn here. And it goes all the way down to here. If it keeps going, it'll leave the muscle, and then it goes up here that way. And all of this allows it to make this branch here, which is the branch to this one neuromuscular junction there. Now, if I were an engineer and I wanted to do this efficiently, I would just make a branch like this, right? So there were things like this. Here's another example. This bifurcation generates this junction. That's fine. But this bifurcation co-fasciculates along these white arrows. And only over here does it peel off and then cross over to that. I mean, that's just wasted stuff. And you may think, oh, these are delay lines. No, the delays here are microseconds, and muscles are hundreds of millisecond contractions, so these delays mean nothing. So and everyone is different. So one of the questions might be, where does all this variability come from? And the answer is, it comes from the fact that in development, that was not the wiring diagram. The wiring diagram in young animals was far more complicated than the wiring diagram in the adult. And I'll just show you that here quickly. At 2 weeks of age, you have basically the adult wiring diagram. One axon comes in and it makes a bunch of neuromuscular junctions. That's just like the ones I was sliding in one at a time. At birth, each of these axons innervates virtually every muscle fiber in the muscle. This is just one axon, but now rather than just contacting a few, it's contacting 85% of the muscle fibers. So there's a dramatic branch pruning that's taking place in life, and it starts at birth. As animals begin to experience the world, there's a massive loss of wires. When it comes to the brain's wiring, it's all downhill after birth. And this is a very scary idea, and this is just different from the way we think of computers. It's basically wired up for everything, and then based on experience, the massive amount of this wiring just disappears. And what this means is that there must be multiple, and so that maybe this is what engrammatization is about. It must mean that each of these neuromuscular junctions must be innervated by many axons at birth, because each axon is doing so much. And here is a three-dimensional reconstruction just through one neuromuscular junction at birth. So that's the muscle fiber that Juan Carlos Tapia painted pink. And here, all these colors are different axons that converge on this one neuromuscular junction. And it's impressive, because there's only going to be 2 weeks later, only one of these axons is going to be there, but every one of these axons, except for one, is actually synapsing with this muscle. They're all talking to the muscle. Some of them have pretty big territories. Some of them have smaller territories. But clearly, at birth, there's lots of opportunity for who stays and who goes. This one, this little pink one, has already left. And over the next 2 weeks, all but one of these will leave. When you get to about 1 week of age, instead of having 10 axons, you only have 2, and they square off against each other very often, where one takes one part of the muscle fiber. The red is the muscle fiber's receptors, and the blue is the other axon on top of the other receptors. And then we can watch, because muscle is so accessible, you can watch the same neuromuscular junction day after day to see what happens. So here's a neuromuscular junction where blue has one territory, yellow has the other at postnatal day 11. We anesthetize the animal, take this picture, sew up the wound, bring the animal back to the cage with its mother. This only takes about 10 minutes. And then a day later, P12, we take another picture. And you can see, if we just go back a day, yellow has gained territory and blue has lost territory. Come back a day later, yellow has gained more territory, blue has lost more territory. Day 14, now it's just the yellow axon, and this branch will totally wither away. Don't worry, this axon has other neuromuscular junctions it keeps, but this neuromuscular junction now is the lifelong partner, this muscle fiber, of that particular axon. So there's a kind of competition between axons for postsynaptic sites. So to summarize all this muscle stuff, you start off with a very complicated wiring diagram where each muscle fiber has lots of inputs converging on each neuromuscular junction, and you end up with something far simpler. Each axon now has chosen lifelong partners, and it is the one and only driver of those target cells, and you have a much simpler wiring diagram, but based on looking at the same muscle over and over again in different animals, this wiring diagram is different in each and every animal. That's muscle, and I guess you might ask, could the same thing that's going on in muscle also be happening in the brain? It's a much harder question in the brain, and we built a machine to cut brains very thin so that we can do the same kind of work, but using not fluorescence, because things are so packed together, we have to use electron microscopy. And this device, which sort of looks like a movie projector, was built by Ken Hayworth and Richard Schleck. It is a device that turns a block of brain into a movie, if you will, a series of frames. And all the action is really up here. All the rest of this is just a tape drive. You have a block of brain that's embedded in a hard resin. It's dead. And then you move the block up and down against a diamond knife, and off comes these very thin sections that are 30 nanometers. That's 1,000th as thick as a hair. So they're virtually invisible, and they come up on this tape. So you end up with the brain on tape. Then you cut the tape into strips, and you put them on silicon wafers that are flat. Each of these is one of those sections. And then you look at them in a scanning electron microscope. I've asked Bobby Kasturi here to hold his hand very still. Daniel Berger made this movie as we move in with the camera closer and closer to one of these sections. At some point, Bobby holds his breath, and we go into the vacuum of the electron microscope, about now, so he's not really holding it anymore. These white things, these big white things are blood vessels. These white objects here are nerve cells. These white stripes are dendrites. These black coated objects are myelinated axons. And as we go up higher and higher in resolution, finally we get to the point where we see vesicle-filled profiles, dendritic spines, and dendrites. So here is a dendrite, dendritic spine, an axon filled with vesicles. So that is a nice thing to do, and as David showed you this morning, you can then reconstruct things. I want to show you that this data set here, so there's the synapse, sits in larger data sets, that is with the electron microscope. This is a large data set that's been aligned with CPUs. So although this looks like nothing is happening, we're slowly moving through a data set of 30 nanometers. And there's one, the size area that I just imaged for you before is that red spot there. Do you see that? Can you see that red spot, or is it invisible? That's how big that last data set was. But what you'd like, this is a cortical thickness, so you'd like to do this over a big area. And this takes time. So we need ways of going fast. We only get about 1 terabyte a day with our single beam scanning microscopes, which means even doing a cubic millimeter, this is about a square millimeter, that's 30,000 sections, would take us 2,000 days, which is too long. So we needed a faster way to go, and the solution to that is this device here. I'm half smiling, and I'm half smiling because I'm afraid of heights and I'm standing on a rolling chair. And also, it's extremely expensive. So this is a multi-beam scanning electron microscope that generates 12 terabytes a day. And it's the Multisem 505, so you know they've been making them a long time. Of course, they haven't. This is the first one ever delivered anywhere. So this name is just there to give me confidence that I didn't waste my money. But it's a brand new machine. And instead of having one beam, it's scanning with 61 beams. And now it's naked, and it's complicated. And the 61 beams are arranged like a shower head, and the one in the center is the one of the 61. So it generates images that instead of being like this, now we get this much larger area made up of a hexagon, and we can tile those together. Then we have to turn that into data, and so we have to color these in. And this is a program, a manual coloring book program that Daniel Berger made called VAST. It's easy to use, and it allows us to trace objects from one section to the next very nicely. And this is a saturated reconstruction. Every neuronal process is labeled there, but it's slow. So we have to get automated, and we're working with Hans-Peter Pfister's group to generate deep learning algorithms that allow us to get fully automated results. The fully automated results, this is a data set that was done entirely by machine and entirely by hand. It's the same data set, and they're rocking back and forth in synchrony. The colors are different, but otherwise they're the same. To a first approximation, this looks excellent. Excellent. This is work Seymour Knowles Barley did when he was in the lab. He's now at Google. But this arrow points to the problem. And when people talk about how smart machines are and how great they do things, just look at the error it makes here. So when this thing starts here, there's a little green process that turns blue and then it turns red. You see that process? Blue, green, blue, red. Can you all see that? No human being would ever make that color. A 4-year-old would not make this problem. And yet we have trouble getting our computers to see the way we see. Ironically, this is visual cortex. So if we wanted to know how to make a better algorithm, all we have to do is trace this at full resolution. The problem is the algorithms to trace it at full resolution require us to know how to see the way humans do. but that data is embedded in here. So we're in an unusual place where the answer is deceptively hard to get at, even though it's right in front of our eyes. But the thing you can do now is, although this is not correct, it has lots of those errors, no human being could trace this amount. And this was done entirely by machine from the Pfister group. So what you end up with is data sets that look like this. These are 2 dendrites with all their spines. And in this little volume we saturated, so we see all the other dendrites in the same data set. That little arrow points to the spine that's sticking out the furthest right there. So there's lots of dendrites in there, but there's 7 times more axons in the same data set. So any gaps you see here are filled by axons. It's kind of hard to believe these things both fit in the same place. You see that hole there has a dendrite, it has an axon, but that's not all. There's also glial cells there, supporting cells. So this is everything. And you know, it's either amazingly depressing or just depressing. It depends on your point of view. There's just 1,600 synapses in this little volume. And even though this is just a little piece of microcosm of brain, there were some interesting regularities. And one of them was the same axon often makes multiple synapses, this blue axon, on the same dendrite, much more than expected by chance. And this, again, may be some kind of strengthening some inputs by, even though this axon's bumping into many dendrites, it's making many of its synapses on one and ignoring many others. The last thing I just want to say is brain networks. We've tried now to look at a brain network. This is work of Josh Morgan. We took the simplest part of the brain, which we go from the retina to the thalamus to the cortex. The thalamus was simple because each type of cell in the retina, all those cells I showed you, has a particular property, and they are known, based on physiology, to drive different types of thalamic cells. So there are these B-type bi-tufted cells and these spherical dendritic cells, and one sort has small synapses and the other sort has large synapses. So we were going to reconstruct this network. And I'm not going to tell you the details of how we did it, but we reconstructed Josh Morgan reconstructed a substantial portion of this and then made a wiring diagram based on the distances. Here is just the strength of the connections between various cells and axons. And I'm just going to cut to the chase. What was observed is, first of all, that there was one network, not a bunch of parallel networks, and it was unexpectedly complicated. Different kinds of retinal ganglion cells innervated individual target cells. So the small and the large synapses ended up on the same kind of target cells. Individual retinal cells that have the same type of synapses innervated different kinds of target cells. And individual retinal cells made different types of synapses on different kinds of target cells. This was all just very complicated and completely counter to what we had expected. We couldn't find any cell types that were predictive of connectivity, for example. So my conclusions are the structure of the mammalian nervous system is self-organizing during development to build a unique brain that is tuned to the environment the animal finds itself in. The tuning is based on extensive competition between the brain's individual cellular elements, each of which seeks to ultimately strongly influence a few other cells rather than weakly interact with many, as they do initially. This reorganization leads to massive simplification of the brain structure as it matures. My children would agree with this. They say, I have become a simpleton as I've gotten older. I only say the same thing. They never ask me questions anymore because they know what I'm going to say. So I think there is some truth to that. Nonetheless, the final networks that we see are inscrutably dense. They're just, as I say, 1 billion synapses per cubic millimeter, and they're very complicated. And nanotechnologies, such as genetic engineering and automated electron microscopy, using also a lot of deep convolutional nets and artificial intelligence in computers, are the tools that allows us to peer deeply into the workings of the greatest nanomachine, the brain. But I'm not sure it brings us very easily to a way to design a machine just like this. So I'll leave it at that and just emphasize that I didn't do any of this work. There were many people who worked on this, many collaborators, especially Hans-Peter Pfister. I want to emphasize him very much because none of the results would have come out without him. And Richard Chalek, who keeps all of these very sophisticated microscopes running and the kindness of strangers, especially IARPA. Thank you very much.